Anyway, it's my pleasure to introduce today's speaker, Megan Greischar. Megan did her undergraduate at Indiana University, double majoring in math and biology with a minor in chemistry. Then went to Penn State University, where she got her PhD in entomology. I'm not sure, she may be the first CAM Colloquium speaker to have a PhD in entomology, but don't let that fool you, because she was working with two of the leading figures in modern infectious disease theory, Otto Bjornstad and Andrew Reed, getting her PhD in 2014, then going on to the University of Toronto, continuing work in infectious diseases. Since July of last year, Megan is an assistant professor in the Department of Ecology and Evolutionary Biology here at Cornell. She is one more speaker out of several in the CAM series to talk about infectious diseases, but it's a different kind of thing. It's not about transmission and population level stuff. It's about the very important things that happen inside the individual who's getting infected and how the parasites do or do not gain the upper hand. You will be telling us today about the impact of ecology across scales on parasite strategies within the host. Megan.
Okay. Well, thank you so much for that very kind introduction. I'm really excited to tell you about my research today. And as Steve mentioned, I do focus on parasite strategies inside the host. And I most often focus on malaria infections because it's a very data-rich system where there's the potential to learn quite a bit. So I'll start by telling you a bit about malaria biology. You may be aware malaria parasites replicate inside the red blood cells of their host. And this replicative cycle plays a large role in determining parasite success within that host. So parasites have to build their numbers, and they're always getting picked off by the host immune system. And I'm not going to have a chance to talk about it today, but a lot of my research focuses on the timing of this replicative cycle. And you might know about malaria is that often this cycle takes place on a circadian rhythm and results in these periodic fevers. And I'd be happy to chat about that with folks who are interested. But I'd actually like to focus on a different part of the malaria biology, and that's the fact that they have to produce specialized forms to transmit to mosquitoes. So parasites invade red blood cells, and some fraction of those invaded red blood cells instead develop into these specialized transmission stages. And those stages contribute nothing to success within the present host, but they're absolutely required for onward transmission to mosquitoes. And so malaria parasites face this kind of interesting allocation problem in their lives within the host. So I use models to try to find context for some of the diversity we see in parasite traits. And I'll tell you about 3 projects today. I'll spend most of my time on the first 2, looking at what parasite strategies can optimize host infectiousness and maximize epidemic size. And then I'll switch gears a bit at the end to talk about some efforts I've made to try to take actual infection data and understand how parasite strategies may be responsive to cues within the host. And so to begin with, I'll talk about a model I developed to try to understand allocation strategies within the host. And that turned out to be a bit complicated, so I had to find some alternatives to some of the standard methods for analyzing it. So as I mentioned, malaria parasites face this allocation problem. And so that means from the perspective of a parasite, each red blood cell is a resource that can be used either for replication. So depending on the species in question, that could be anywhere from 0 to 32 new infected red blood cells from that single infected red blood cell. Depending on whether there are red blood cells available to infect. And so that's a huge opportunity to expand parasite numbers within the present host. And alternatively, that red blood cell can be used to make one of these transmission stages, which is a chance at infecting the mosquito. And biologists have been quite intrigued about how parasites trade off between these 2 parts of their life cycle. And that's because this replicative cycle is responsible for all of the human misery we associate with malaria. So we'd like to get a better handle on how parasites are balancing this trade-off. And I find it an interesting problem because it's also an iterated allocation problem through time. So parasites have to make an iterated series of allocation decisions, and what they do early on may influence what they're able to do later on. I built a within-host model to encapsulate this allocation problem, and I'll just walk you through what that looks like. So I think of the host as kind of a bag of blood, is how my advisor Ottar always described it. So basically, there's a population of uninfected red blood cells within the host. They're replenished in a logistic fashion if they're removed. And so the idea is that the host will maintain this homeostatic equilibrium K. And so there's some background removal of red blood cells that happens naturally, but in the absence of infections, host will maintain an equilibrium level of red blood cells for normal functioning. But then there's also the possibility that red blood cells can be lost due to parasitic invasion. And there are these red blood cell invasive forms called merozoites. I assume that they invade red blood cells at this constant rate P. And from there, a red blood cell is lost from this uninfected compartment and enters the infected compartment. And so there are a couple key features to this cycle of replication within the host. One is that I've modeled it with a fixed time delay. And so that's kind of an important feature. So a standard method might be to use an ordinary differential equation model. So there would be some constant rate at which parasites are maturing after invasion. But that yields an exponential distribution of waiting times during this replicative cycle. And biologically, that means that sometimes parasites are multiplying, bursting out of red blood cells immediately after invasion, which is biologically unrealistic, but more importantly, if I assumed that, it would dramatically overestimate parasites' replicative capacity. And I want to get a handle on how they trade off between these parts of their life cycle. So I've modeled that with a fixed time delay. And then there's some fraction of these invaded red blood cells that will go down this different developmental pathway. And the other thing I should mention here, you might be wondering why I'm using X to denote time instead of T. That turns out to be important later when I look at a model across scales, because we'll have time running at two different scales. So X indicates the age of the infection, so the time, the days following inoculation. And later there will be time in the normal sense. Okay. And so this first term is the recruitment of infected red blood cells. So that's pretty standard. Just the fraction that aren't allocated to transmission will continue in this class. They can be removed at this constant background rate. I've also explored what happens when host immunity is removing red blood cells, for example, in a density-dependent fashion. So the model can be expanded to include that. And then because this is a fixed delay model, the cells that exit this life stage are the ones that were recruited exactly alpha days ago, depending on whatever level of transmission investment occurred alpha days ago. And then not all of them will survive that delay, so this exponential term captures that. And then this merozoite class, these are the ones that burst out of the infected red blood cells, and there's an expansion here. So omega daughter cells burst from each infected red blood cell. And I'm assuming that the merozoite lifespan is exponentially distributed in the absence of invasion. And that follows from some work looking at parasites in the Petri dish. And they die at quite a fast rate, actually, like minutes. Whereas the length of this full replicative cycle, so here I'll be looking at a rodent malaria parasite, and that cycle takes 1 day. So merozoites are lost quite quickly. And then they can also be used up, of course, if they invade red blood cells, then they're no longer able to infect more red blood cells. And so now moving to the transmission part of the life cycle, some fraction of the parasites are allocated to become immature gametocytes. They're removed at a background rate, and then there's this additional time delay. So I think this is an important characterization, important feature of the life cycle. Whatever allocation decisions are made, there's going to be a time lag before any benefits can be realized from that. And in this case, it'll be about 48 hours, but for some malaria species, that could be like a week. So many cycles of replication could happen while these immature gametocytes are waiting to mature. So this is the same kind of formulation as before. And then these are the mature transmission-ready gametocytes, the transmission stages that can be passed on to mosquitoes, and they die at a different rate, mu sub g. And some past theory has looked at how should parasites allocate in order to maximize their total production of transmission stages. But I wanted to go a step further here and account for the fact that not all of these transmission stages are created equal when it comes to infecting mosquitoes. And in particular, there's density dependence here. So this is a curve generated from some experiments in rodent malaria, where this is the number of transmission stages in the blood and the fraction of mosquitoes that get infected if they feed on that host. So this is what I'll be calling beta, the level of host infectiousness. And it's the particulars vary a bit for different strains, but it's always the sigmoidal shape. And so that means there are accelerating benefits for parasites to make more transmission stages when those transmission stages are rare. But if there are many transmission stages around, the benefits of making more are negligible. And so I wanted to incorporate that. Yes, and there's a question.
I'm sorry, if you could go back to your previous slide. Mm-hmm. It's a complex model, and I'm trying to keep track of how different pieces are interacting with each other. So I with a subscript G, do they become G?
Yes, unless they die first.
So I'm looking for the equation that gives you rate of change for G, and I sort of expected to see I sub G on the right-hand side. And
yeah, so I did this for clarity, but possibly made it more confusing. I didn't actually have to I didn't have to write this out with the IG term explicitly in there. It's actually just the number of cells that entered that developing class alpha G time units ago.
I see.
Yeah, so the IG class doesn't figure explicitly into the gametocyte equation.
I see. Thank you very much. Sorry to interrupt.
No problem. Thanks for asking. Okay. So translating from the transmission stage abundance to the fraction of mosquitoes infected. So that's just an experimentally derived curve. So it's maybe not a very pretty form, but that's what I have to work with. And so from that, I can ask what strategy would maximize the cumulative host infectiousness over the lifespan of an infection, where A would be the maximum lifespan of an infection. And so altogether, this is the model to work with. And there are a lot of moving parts. So there are multiple time delays here. There are multiple density dependent interactions. And so that rendered the more typical optimal control methods intractable. And so I've used instead numerical approaches to try to understand the system. And so these are simulations for 3 different levels of transmission investment. So the purple is a suboptimal level of transmission investment. The black is optimal. And then this light gray line is super optimal. And so this is just the abundance of infected red blood cells. And so you can see that the transmission allocation, and I possibly forgot to mention, but this is important. For this first step, I'm assuming that allocation is constant over the lifespan of an infection. So it's a fixed level, a fixed percentage of infected red blood cells developing into these transmission stages. And so these are the infected red blood cell abundance, and this is the transmission stage abundance. And so because it's a fixed level, the trajectories roughly follow each other. except you'll notice the transmission stage has these jaggedy bits. That's because the mortality rate is rather high for these transmission stages. But the key thing to notice here is that allocating to transmission actually slows down the growth of the infection. And that's to be expected. So it's diverting resources away from parasite replication. And so that means that parasites hit a later peak and a lower peak in their infected red blood cell abundance. And that carries over into the transmission stage abundance. So investing more into transmission actually means that the host is going to be infectious later. And we can translate that through to this host infectiousness. And we see that if there's a low amount invested into transmission, this purple line, the host becomes highly infectious very rapidly. But the parasites actually kind of overexploit host resources. So there's a dramatic crash in their numbers. And that's reflected in this host infectiousness. Versus the optimal level is a bit slower, but it reaches a high level of infectiousness and maintains that for longer. And then the super optimal strategy just delays things too long. And so that kind of explains why there's this optimal fixed strategy. And I can see the fitness landscape. I can visualize the fitness landscape. So this is the level of transmission investment, again, fixed over the lifespan of an infection. And this is the cumulative level of host infectiousness for an infection that's lasting 20 days. And the optimum happens to fall near 42%. And there are various aspects of within host ecology that can shift that optimum one way or another, including immunity. But I then wanted to know how should the best strategy vary through time? And so that would be an ideal place for optimal control. But instead, I used a spline to specify transmission investment. And so this is the spline here. So I specify a spline with few parameter values. That's the nice part about splines over days post-infection. And this is the level of transmission investment. And you can see here the cumulative host infectiousness. And I set this up as a Nelder-Mead optimization. And it's just figuring out what spline parameters will give us a strategy that maximizes host infectiousness. And so you'll see it's still wiggling. My initial guess was quite poor, so that started with a high level of allocation and decreasing. There are dramatic fitness gains for restraining investment early, so allowing parasites to grow up to large numbers before there's appreciable investment into transmission. And so when it's finished, it'll look very similar to that. I can compare, this was the best fixed strategy at 42%. And then the best time varying strategy looks more like this. And so these early investment decisions can constrain the future options. So for this fixed strategy, the infection grows much more slowly than it could. And I did some other work modifying this best fixed strategy to try to understand why parasites should scale back their investment here. And that turns out to happen because resources have become limiting. And so the parasite population is going to crash. And if parasites continue to allocate at a high rate, the population will crash much more severely. Versus if parasites can invest in growth, they can soften the decline a little bit. And so that was a single infection case. I then wanted to examine what if there's co-infection. And so this is two strains infecting the same host at the same time. And so I'm assuming that they're identical, except for possibly their transmission investment. And so that changes the objective function in a kind of interesting way. And so if this is the cumulative infectiousness for a focal strain, the first kind of interesting thing is that more of these transmission stages are better from the perspective of infecting a mosquito. So more gametocytes are better, and that means the focal strain benefits from having a competitor present within the same host. But at the same time, each strain wants to maximize its representation in the blood meal that goes to a mosquito. And so there's this kind of tension where the competitor can be useful, but is also disadvantageous. And so from that, I wanted to ask, what's the best fixed strategy? And so this is something of a pairwise invasibility plot. And I just numerically went through and figured out the level of transmission investment for the focal strain on the x-axis and the investment of the competitor on the y-axis. And the colors here represent the relative fitness of the focal strain, meaning in this gray to black region, that's where the focal strain is doing better. And the red-orange regions are where the competitor is doing better. And so basically this suggests selection should push the system towards this point here, which falls at approximately 10% investment. And so that means over here, for example, the focal strain can improve its relative fitness by increasing its transmission investment. And over here it can improve its fitness by reducing it. And conversely, in this part of the parameter space, the competing strain could improve its lot by reducing its fitness. And so we would expect the system to be pushed towards this evolutionarily stable strategy. And so we would expect if a population of parasites were using this strategy, it could not be invaded by a mutant strategy. And so this suggests that co-infections select for greater replication at the expense of transmission. So 10% is a lot less than 42%, which was the best single infection strategy. And so this is another mechanism by which, and there are many that come up in evolutionary biology, whereby co-infections might select for nastier parasites in some sense. So these parasites should replicate faster than they would otherwise do. To put that another way, it's basically a tragedy of the commons type scenario. So each strain, because the competitor is present, benefits from replicating faster than it would otherwise do. Okay. All right, and then I wanted to know, the last piece of this study was to try to understand what if these co-infecting strains can use strategies that vary through time rather than these fixed strategies. And so that's something I called dueling splines, basically. And so I started with this best single infection strategy from before, and then put it into a co-infection and looked at optimize the focal strain to figure out what is the best response to this single infection strategy. And as you might imagine from the previous results, the best response is to pretty dramatically reduce investment into transmission. So parasites have to replicate faster. And then I iterated this process. So then I assume the competitor is using what was previously the best response strategy and ask, what is the strategy that is now the best response to that? And so I iterated this process, and eventually the splines settled on this strategy. And so that's a candidate evolutionarily stable strategy for co-infections. And it, again, is suggestive that co-infections select for more replication at the expense of transmission stage production than we would see in single infections. Okay, I'm going to pause for a moment in case there are any questions.
If I may again.
Sure.
So this is a graph of C of X, right?
Yes.
Yeah. Is it something which you can actually measure in practice to see what they do?
Yes. Yes, that's the last section of this. So yeah, yeah, good question. Yeah.
And here there is no real difference between the focal strain and whatever is the other one.
The competitor, yeah.
The competitor, their payoffs are symmetric, right?
Yes.
This is why you're looking at the SS.
Yes, exactly.
I see. So I was a little bit surprised to see the shape of the saddle which you were showing, because there wasn't symmetry there.
You mean in the fixed?
Mm-hmm.
You said you were surprised because there was not? Sorry, I'm not sure I understand your question.
Oh, I think I misinterpreted this slide. Thank you.
Okay.
I now understand what's going on.
Yeah, so it's not a, sorry, I should have been clear that it's not a saddle point. It's just, yeah, where I would expect selective pressures to push the system over time.
Mm-hmm. Thank you.
Sure. Okay. So it looks like time varying strategies can really help parasites optimize their host infectiousness. And I was able to use splines as kind of an alternative to optimal control in a system that had a lot of moving parts. So I'll now go through what allocation strategy might maximize epidemic size. And for this, because I'm increasing the complexity at the larger scale, I'm going to reduce complexity in the within host model and just look at fixed strategies through time. And so there's a few different ways I think ecology outside the host should matter. So first of all, before I was implicitly assuming that infectiousness is equally valuable at every point during infection. But we know that shouldn't be true. So hosts recover. So if you make a host infectious very late, that might be useless from the parasite's perspective. Secondly, mosquito populations can change dramatically over the lifespan of an infection. And then finally, epidemics grow and decline. And so that changes the age distribution of the infections themselves. And so that should make different parts of the infection more valuable than other parts. And so I wanted to understand how these processes would interact to select on allocation to transmission. And so I'll focus first on the recovery bit. So I reparameterized my model from before for human infections. So the numbers might look slightly different, but it's the same model. And the same result holds as before, that transmission investment delays host infectiousness. And so here you see 5% transmission investment makes the host rapidly infectious, but it settles down at a lower equilibrium level of infectiousness, versus 40% takes a while to make the host infectious, but it ultimately equilibrates to a higher rate, a higher level of infectiousness. And here, because we're going to have transmission from mosquitoes, which is another beta term, these betas will be host to vector, so sub-HV. And so I first wanted to know for these parameter values. Oh, sorry. Yes, there's a question.
Yes, on the graph that you just showed, I'm wondering where we get these very small oscillations in the graph of beta sub HV. Is there sort of like a physical understanding of where those oscillations would come from?
Yeah, so this is, yeah, that's partly related to the mortality rate for the transmission stages that can be more rapid than for other cell types. So that's part of it. Some oscillations are also due to the fact that I started these infections with a uniform distribution of parasite ages, like the infected red blood cells were a uniform distribution, and that's not the equilibrium stage distribution for this model. So there's some oscillations related to that.
Okay, thank you.
Mm-hmm. Okay. And so I wanted to know what transmission investment level will maximize host infectiousness over this period. And here, 280 is the maximum length of infections we considered here, which is reasonable for human malaria infections. And the answer turns out to be 20%, which is obviously sensitive to the parameter choices, but it serves as a baseline. And so I then wanted to layer in recovery. And so this gray curve would be the fraction of infections persisting. And I'm assuming here just an exponential distribution of waiting times with a constant recovery rate. And I'll come back to that. It might seem weird to have the recovery rate be completely decoupled from the within host dynamics, but that's the simplifying assumption I'm going to start with. And so if we layer in that recovery rate, you can see that's just the joint area under this curve. And you can see how that would make it even more important to render the host infectious rapidly. And so when we look, that does constrain the optimal level of transmission investment. So it's a lower level because these later time points are less important than they would otherwise have been. And so we can consider, as recovery rate changes, what is the optimal level of transmission investment I'll just summarize it here. So at the top is just that single infection optimum I started with no kind of epidemiology in the model. And if we layer on any recovery rate, that reduces the optimal level of transmission investment. And as the recovery rate is faster, that reduces it still further. And so I then wanted to understand how mosquito availability might factor into all of this. So I teamed up with Lindsay Beck Johnson, who developed a really nice model of mosquito population dynamics as a function of the amount of larval habitat that's available. So mosquitoes, maybe I'm showing my theoretical entomology background here, but mosquitoes mature in aquatic habitat. These immature stages are not directly relevant to malaria transmission, but interactions in this state can determine how many adults will emerge, and that's certainly relevant to malaria transmission. And so I've put the references there if you're interested. She has a really nice delay differential equation model that runs on temperature and determines, helps us understand the number of mosquitoes that should be emerging under different temperature regimes. And so the idea is that susceptible vectors will be recruited from their larval habitat at this rate R sub A. And when they're recruited, they'll be recruited as susceptible uninfected vectors. And I can modify the mosquito availability by changing the amount of larval habitat. So it might be increasing, for example, if a rainy season is starting, or it might be decreasing, say as a rainy season is ending, or it could be constant through time. And vectors enter the exposed class at this R sub VE term that depends on the biting rate as well as the host infectiousness, which is a function of host age. So that's why I have this X, a function of infection age, not the age of the host. So that's why this beta term is a function of X, but this R sub VE is a function of T, as in like the time in the season. And this is what the term looks like. So susceptible vectors have some bites that they allocate to N hosts. And some fraction of those hosts then will be in this infected class. For ease of comparison with the previous results, I assume that all hosts begin the simulation with infections age 0 and then progress. And they recover at this constant rate psi, and they are infectious based on this beta sub HV term. And that's when T is less than this maximum infection age. And then once we're past that age, no new vectors can be infected by the focal hosts that started the simulation. And so there's another important delay here, and that's when mosquitoes are exposed to infection. There's this extrinsic incubation period, which I denote tau EIP, before they can actually become infectious. And so if we want to understand the allocation strategy that maximizes host infectiousness, that's the one that maximizes vector exposure, tau EIP days ago. And then again, there's this survival term, and that's especially important here because it's actually the case that most vectors won't survive this incubation period. And so I wanted to understand what strategy maximizes the recruitment of infectious vectors. And so if mosquito numbers are constant through time, that's basically the same question we were asking before, so we'd expect to get the same answer back of 12% for this recovery rate. If mosquito numbers are declining through time, that should make it even more important to get the host infectious early, so we would expect that to reduce the optimal level of transmission investment. And conversely, if mosquito populations are expanding, that makes it more important to have the host infectious at these later time points, even if it's pretty unlikely that the host will get there. And so we would expect a greater level of transmission investment then. And so we find for constant mosquito sizes, we get back indeed the same answer as before. If mosquito populations are shrinking, that's this minus sign here, we get a reduction as expected in the optimal level of transmission investment because the parasites have to replicate faster to make the host infectious rapidly. If mosquito numbers are expanding, indicated with a plus, that increases the benefit of this later infectiousness and should increase the optimal level of transmission investment. And then finally, well, I'll pause for just a moment there in case there are questions on that. Okay. So the final piece of this is that epidemics grow and decline. And so to understand that, I worked with Nicole Medeo at the University of Toronto to embed all of this in an epidemiological model, which looks quite pretty here, but each of the, like the RHR terms, are quite ugly. So I won't show them to you now because there's a lot of, well, there's a lot of time delays and convolutions in here. But the key question is, what parasite allocation strategy would maximize recruitment of infected hosts? And this infected term is relatively straightforward. Just the number of susceptible hosts that come into contact with infectious vectors, which bite at this rate B, and then the vectors transmit at this beta sub VH term. And so we can think about what that might do to the optimal strategy. And it's important to consider how epidemics alter the mean age of infection. So as an epidemic is expanding, most of the infections are in the early stages. So that means that biases the age of infection towards these early time points. And conversely, if an epidemic is declining, most of the infections are old because the infections are not on average replacing themselves. So that biases the age of infection to these later time points. So we'd expect transmission to be, transmission from early time points to be more important here, and transmission from later time points to be more important here. And so for the parameter values I'm showing, I'm basically capturing this first part of the epidemic curve. So the function of adding all of this epidemiology here is basically to bias the infections towards these early time points. And so if we look at what that does, it tends to bring down the optimal level of transmission investment. And again, that's because parasites have to make the host more infectious rapidly. So they have to scale back this level of investment. And the pluses and minuses, again, indicate different mosquito dynamics. But for this case, it doesn't matter much. Whenever we layer in the epidemiology, it reduces the optimal level of transmission investment. And so I wanted to understand all of this complicated ecology. How does it stack up against a selection pressure we know is pretty important, which is the presence of a co-infecting strain inside the host? And so for the same system, I asked, what is the optimal level of transmission investment in a co-infection? And that turns out to be down here. So this is a huge selective pressure on transmission investment, and it seems to dwarf all of this external ecology, which I found to be a bit of a bummer. But I wanted to revisit an assumption I made early on. So in particular, I assumed that the host recovery rate was independent of any dynamics happening within the host, and that need not be the case. Unfortunately, we don't really know what the true case is. So it could be, we would expect the level of transmission investment to influence, but we don't necessarily know how. So I tried a couple different ways. So I first assumed that recovery rates would be lower as transmission rate increases. And so that could happen if parasites are able to avoid the immune system by growing at a slower rate. And so if we try those dynamics, that increases the optimal level of transmission investment quite substantially. So essentially here, higher levels of transmission investment make the host infectious late and also increases the likelihood that the host will get to those later time points. And so this is the optimal strategy here. And we could also consider the case, because we don't know what the true pattern is, where transmission investment actually hastens recovery. So if parasites grow too slowly, they might be removed more efficiently by immunity. And if that's the case, that can dramatically reduce the level of transmission investment close to what we would get with the presence of a co-infecting strain. And so if we allow for interactions between within host processes and the recovery rate, that means that this external ecology could be very important to determining parasite strategies. And so it really bothers me that we don't know which way the pattern goes. So some of my current work is focused on looking at some human time series. And this is in collaboration with Lauren Childs to try to figure out how transmission investment might alter the duration of infection. And so these are human infections. This is called the malaria therapy data. And so this was before antibiotics, they actually, the best treatment for neurosyphilis was to deliberately infect people with malaria, and it would cause fevers that would get rid of the neurosyphilis. But they were able to drug treat the malaria infections. So these are time series obtained in the course of giving patients as good care as could be managed at that time. And we can use these time series to estimate the level of transmission investment and using some of the tools for function valued traits to try to understand how those alter the duration of infection. So I hope to have answers on that for you soon. And so to sum up this section, it's clear that dynamics across multiple different scales can influence parasite strategies. And I'll just briefly end by talking about using actual data to try to understand what cues parasites might respond to. And so people take this sort of time series data, especially from experimental rodent malaria infections. So that's a highly controlled system. There's a lot of really exciting time series that can be used to estimate transmission investment. And I wanted to understand what cues parasites might be responding to in their actual strategies. And one cue we would expect them to respond to is the availability of resources. So if there are a lot of red blood cells around, we might expect parasites to do something very different than if red blood cells are limited. And so people had reported that transmission investment increased with resource availability. Other people had reported that it actually decreases. And both of these studies looked at the same time series data and drew opposite conclusions. And so that made me wonder about the methods that people actually use to estimate transmission investment. And so I have this within host model. And so I used it to simulate data and then apply some of these methods to figure out which one is actually right. And it turned out that none of them were. So here, this is the actual level of transmission investment. I fixed it at 5% over days post-infection. And then I tried a bunch of different methods in the literature and a simple one that I thought might perform well. And all of them show spurious changes in the transmission investment. So we would look at this and think that parasites were doing something sophisticated and interesting when actually they're not. So these are just simulated data, to be clear, but we would like to be able to draw the true pattern from these time series data. And the problem turned out to be a classic one in time series data, and that's temporal autocorrelation. So when people infer transmission stages, they look at the number of these transmission stages that are around, and then how many replicating parasites were around previously. And this is just a simple cartoon of what that looks like. So in this cartoon, the actual transmission investment is 50% constant through time. And so people would take, so here's 3 gametocytes here and divide it by the number of replicating forms previously, that's 4, and they would erroneously think that 75% of the parasites were allocated to transmission. And so the problem is that not all of the gametocytes, the transmission stages here, came from the allocation decision we're interested in. They tend to build up over time, and that creates pileups that look like sophisticated strategies when nothing of the sort is happening. And so we can't separate newly formed transmission stages under the microscope, but we can separate them statistically. So basically, this model looks at the number of gametocytes observed at a particular point in time, and as a function of the number of red blood cells that would be invaded some time steps previously. I estimated transmission investment, again, with a spline. But the key point here is that there's some fraction of gametocytes carried over from the previous iterative cycles. And so this carryover can account for the temporal autocorrelation. And when I apply that to my simulated data, it returns a much clearer answer that shows that in the simulated data, parasites were not doing anything interesting. And if you're wondering why it's lower, that's because gametocytes die before they're sampled. And so I applied that to real data and was able to see that parasites do indeed exhibit time varying strategies. Um, and I wanted to know what within host cues actually trigger these responses. And so to do that, I teamed up with Petra Schneider, and she had some data on rodent malaria infections that were drug treated. And so there were two important places for models in this study. One was figuring out what would it look like if parasites were responding in an adaptive way. And secondly, using these new statistical methods to figure out what are the actual strategies. And so this is the model prediction. Basically, that parasites should invest a lot when things are good. They should scale things back when things get rough, when drugs are applied. And then at some point, things are so bad, they should just terminally invest into making these transmission stages. And so we found a pattern that was roughly the same. We got this asymmetrical U shape by this new approach. And we were also able to figure out that parasites are responding to multiple cues. So they have a different reaction norm depending on what's happening with the uninfected red blood cells. And there were some intriguing differences across different parasite strains. And so this was a particular snapshot of parasite responses at a single point in time, but I'd like to look over the entire course of the infection. So I think in the interest of time, I'll skip this next slide. But I'd be happy to talk about ways I envision that happening if anybody's interested. And so the key point here is that it looks like parasite strategies are responsive to multiple cues, and we now have statistical methods that are capable of detecting that. And so more broadly, I'd like to understand why parasites exhibit such variable strategies. So I use models to give context to that. And I'd like to expand further to try to understand, in particular, some of the dynamics here. These are each different parasite strains. Some are quite repeatable, while the others are quite noisy. So I think this is suggestive that it would be helpful to expand beyond some of these deterministic models in determining what the best parasite strategies are. And that might finally give us a handle on why we see such diversity in the allocation patterns. And with that, I'd like to thank funding sources, and I'd be happy to take any questions.
Thank you, Megan. I'd like to invite everyone to unmute and give Megan a round of applause that she can hear, even though we're at a distance. And let's take questions. Raise your hands, and I will. I'll call on people. Hopefully, I'll get it in order. I'll start this. I'll start it out with a question, Megan. And a lot of the optimization problems that you pose, you're looking at the total amounts of some good thing, like how many mosquitoes you infect, or the total transmission or number of infected hosts, but not worried about the timing. Mm-hmm. And this makes me think about that, the classic question in life history theory of whether you're maximizing lambda or R0, right? Yeah. Is there some, right? If you get a payoff sooner, if you infect a host sooner, and that host can then infect others, is an early infected host more valuable? So what's the? I guess how does timing play into this? Not just the amounts of mosquitoes or hosts that you infect.
Yeah. Well, I guess the answer, as always, is that it depends. But I think some things it depends on are. I think malaria and most diseases would be maintained in a metapopulation sense. So it's probably winking in and out of different populations. So I think what really matters is maybe not the timing of infecting hosts, but whether you can infect a host that's going to seed infections in a new population. And also, there's oftentimes a dry season where transmission is impossible because there aren't mosquitoes around. So I think we also thought about this, but haven't really addressed it yet. What would be strategies that would maximize the odds? I guess our thinking was if you maximize the total number of hosts infected, that probably does maximizes the chances that one of them, at least, will carry the infection through the dry season to when there are more mosquitoes available. But I think the spatial aspect could be important, and we just haven't gotten to it yet. I see a few different questions.
I'm not seeing hands. I wonder what's happening. Why I'm not doing.
Oh.
Maybe you had better take over because I'm not seeing the raised hands thing. Megan.
If you, I have my hand raised.
Okay.
We hear a lot about evolution of COVID these days. How quickly do the malaria parasites evolve?
Yeah.
And is there also evolution in the mosquitoes that affects the dynamics of this whole process?
Yeah, so in answer to the first question, the parasites do, they do mutate and evolve much faster than we would like, although I think substantially slower than COVID. But there's continued problems with the evolution of drug resistance to every anti-malarial that has come on the market, including, unfortunately, the current frontline drug. So there's a lot of evolution there. And then as far as the mosquitoes, there's also way more evolution than we would like. So there's a lot of evolution of insecticide resistance and potentially also evolution of behavior where the mosquitoes are biting earlier in the day so that it can access people who are protected by bed nets at night. So there's evolution at multiple scales here, and it definitely does alter dynamics.
And when you look in the models, can you build in the effects of this by assigning different parameters to different strains?
Potentially. I think the key part that we are often missing is that there are quite possibly trade-offs. So perhaps like a drug-resistant parasite is not equivalent in terms of like replicative capacity to one that is sensitive to drugs, but we don't always know how those trade-offs are, if those trade-offs are at play, and if they are, which direction it goes. So I think that's a necessary component. And ditto for mosquitoes. Sometimes insecticide resistance could come at a cost. And so I think it would be important to incorporate those costs to get a good handle on that.
Thank you.
Sure.
Again, I'm not seeing raised hands. I don't have access to that. So Megan is going to have to, or you're just going to have to speak up.
Alex, I think your hand was before mine.
That's okay. Since I asked before, why don't you go ahead?
So I have a question that's in some ways related to John's. It seems that it's likely that in many cases you might have mixed populations, or even if you're within a single population, I can imagine that some of these timescales, for example, might vary a little bit from case to case. And so my question was, if there is variation in some of these parameters across the population, so that you have not a single time scale, but a distribution over time scales, or some of the other constants are not a single number, but a distribution depending on which type of mosquito happens to be feeding. How much does that change the dynamics? And has there been any work on sort of the uncertainty quantification in the face of that type of uncertainty?
No. So honestly, it was quite difficult to parameterize. It was quite difficult to find data to parameterize this model, even with these constants, which, as you point out, are a very coarse measure of the actual variability that is at play. So we had trouble. Often, like this is quite a basic parameter, but it's often not even known, like how many mosquitoes per person are there in a particular location. And so that's, I would suspect, maybe more important. Like if we can't nail that down, I think we're unlikely to get traction in some of these other things, which are probably also important in terms of, I don't know if you're thinking of variability in biting rates. That's certainly an issue. I also worked with a grad student and now postdoc Tsukushi Kamiya on a project to look at variability in the extrinsic incubation period. And there's known to be variability there, and that is hugely important for the epidemiology of the system. And so that's something we haven't incorporated here, but potentially could in the future. I don't know if that answered all of the parts of your question.
I think that answered the majority of them. I could keep asking more, but I'll let Alex go.
So I have a very basic question. At the very base level, what you have is a system of delay differential equations with at least 2 different delays. I think I see 3 different delays in the current cross scales model. And then you have parameterized time-dependent control, which you are optimizing those parameters, treating it as a black box optimization problem, right? Using the spline parameterization of your time-dependent control. And this is based on forward integration of the system of delay differential equations. When you're starting a system of DDEs, you need to provide the history.
Yeah. So
it's not enough to provide an initial condition. And I don't know if there is some biologically obvious way to get that history here. If there isn't, oh, there is?
Well, I can tell you what we did, but finish asking your question.
If there isn't, how do you know that what you consider to be optimal is not heavily impacted by the history which you used in your simulation?
Yeah. So in the interest of time, I did not talk about the initial history, but that is an important component. I didn't, we thought about it heavily with the cross scales model. So just to be clear, that was a fixed strategy. And mainly when we did thought about the initial history here, we made it as simple as possible so that we could compare to the strategies that optimize fitness at other scales, if that makes sense. So like as in, if we're maximizing the number of infectious vectors, the hosts that were inoculated into that system with infections would start at age 0 for their infections, so that we could compare it with the first case. And in the earlier case, where we did have the splines, I did actually run some simulations. So this is the other part of my research, is thinking a lot about those initial history functions and the synchrony of the replicative cycle. And so if the initial history is quite a narrow age distribution, that would be a highly synchronized parasite development. Versus if it's poorly synchronized, that would be like a uniform distribution. And so for the for computational efficiency, I did a uniform distribution for most of that, but I did also try it with a highly synchronized, a very narrow age distribution, and I got back about like the same answer. So I don't think it's particularly sensitive to that. But I definitely take your point that it could have been.
I see. Thank you.
Okay, we are at time, so I invite everyone to unmute, give Megan a final round of thanks for her talk. And unfortunately, we don't have GatherTown access anymore. For reasons you can talk to David about, I guess. But everyone who is interested in hanging out to chat or asking more questions is welcome to stay here on Zoom for a bit longer. So thank you all for your attendance, and we'll see you next time.
Thanks, everyone.
All right.
We may be down to the last couple.
Yeah, no worries.
I'm just hanging around in case someone asks a good question that I didn't think of.
I mean, one of the sort of a philosophical question, and this is me mostly working with physical systems, maybe, you know, it seems like there's a huge amount of uncertainty in what sort of the right models are in some cases. And, you know, and there's much less in the sense of, you know, there's a conservation. Okay, so there's some type of conservation law, right? That if you know, if things start just magically appearing, there's probably something wrong. But there's much less of a sense that there's strong constraints based on physical principles that we really understand, and there's much more empirical modeling. I guess one of the things that I just don't have a great sense for is how do people evaluate, you know, is this a good model or not, or is this a reasonable model? How do you go about saying, you know, I think there's something that really needs a great deal of improvement and it's here rather than here?
Yeah, so so in a lot of the epidemiological models, people have moved to ensemble modeling methods. So trying to, ideally, you would have like completely different groups working on the model and not talking to each other and making different assumptions and parameterizing the models differently. And then actually, Lindsay Beck-Johnson, who I mentioned, has been working with a lot of those ensemble models for foot and mouth disease to try to, because, you know, it's a pretty urgent practical problem to try to forecast these infectious diseases. And you're right that often we don't even know like what the appropriate model framework is, like much less the parameter uncertainty. And so she speaks quite highly of those approaches. So I think that can be a good way of, you don't necessarily, it kind of brought home to me that you don't necessarily need a model that's right. You just need to know how it's wrong. And if you compare it, like it's possible through ensemble methods to kind of get a handle on that.
Right. I mean, maybe a related question is, you know, for a lot of complicated models, there's often a hidden, much simpler model that captures a large amount of the dynamics correctly. You know, which sort of have always found interesting, both from my deterministic modeling hat, right? So this notion that if you give me a model that I actually understand, often there's a reduced order model that does a pretty good job of capturing the dynamics that you see, or, you know, Jim Sethna talks about sloppy models where there are some parameters that it just doesn't really matter how you set them, you get more or less the same dynamics, which I think reflects the same thing. It's also interesting to me from this data-driven perspective that, you know, people think about sort of very flexible model classes now, where you try and put in some knowledge of what reasonable assumptions about the shape of the model might be, but there's a lot of model parameters and you just try and run it on data and train and see what happens. I guess one of the things I don't have a sense for is, for a model that's sort of as complicated as the system of delay differential equations that you were looking at, even independent of how accurately it captures the system dynamics that you care about, do you have a sense for how sensitive it is to sort of errors in each of those terms, just what the internal consistency is, or could there be terms that would be dropped and give more or less the same dynamics, or is that known?
Yeah, that's known to some extent. So some of the bit, like the slide I skipped over, that was some work with Tsukushi Kamiya. So he's used, I think, some really nice methods to try to get a handle on that. And he had like a semi-phenomenological model, a network model of host immunity. And so he could see which components are actually necessary to include, like what do they actually add. He also got a really nice handle on like covariant parameters, like where there's not like, and you know, as frequently crops up that you can't independently estimate these parameters that have distinct biological significance. And so he used some really nice hierarchical Bayesian methods to get a handle on that but I think all of that relies on, like he got that to run on very detailed time series data from these rodent malaria infections. And so, yeah, stuff that we just couldn't possibly have for human infections. So I'm not sure what that says about our ability to do this in human infections, but you have to imagine if the rodents are noisy, the humans must be, way noisier.
Yeah. Although, I mean, one of the things that I think is interesting too is that some of the questions at this level have less to do with the how well the model fits the data and more to do with sort of what set of dynamics the model can even express, and whether simpler models can express the same dynamics or not. And sort of how sensitive those dynamics are to all of the different parameters, of course. I guess part of the reason that I'm thinking about this is, that type of question about how do you look at a data generating process, which may be purely computational, and internally say, is there a simplified process that captures the same dynamics, at least for some regime? That's exactly the type of thing that would make a good class project for. It's so the section on dynamics for my class is the last 2 weeks, right? So it's not like they'll have a lot about fitting system dynamics earlier in the semester, but nonetheless, I think it's something that I've been kind of interested in and could be fun to talk about further.
Yeah, definitely. Yeah, I admit I haven't given as much thought to that as I could have, because honestly, this was like the simplest model I could come up with that I thought would do the job. And it's already like way too complicated. So
exactly. It's totally reasonable. I mean, I guess I got involved in some of these, so I've been interested for a while in, I have a long-standing connection to controls, and in particular the use of model order reduction techniques for controls and for understanding engineered systems. Usually much simpler systems than these, right? So it's frequently linear dynamical systems, and or things that there's not a linear dynamical system, but keeping a minor amount of nonlinearity and history variables gives you sort of a rich enough class of models that you can capture a lot of things. But partly because of looking at some of the things around power grid dynamics, I started to get more interested in constrained nonlinear models and non-constrained nonlinear models. A lot of the model order reduction stuff has been moving in this direction too. And so, some of the things, this question about, do you start with a system of equations and try to find a good set of reduced variables, for example, that will capture the behavior, right? Which would usually be like a PDE model then, rather than something like the ODE models that you're working with. But even so, like looking for, what are the dominant nodes, or what are the low dimensional subspaces that you explore in your dynamics? That's been kind of interesting, but it's been interesting too to watch the evolution of the data-driven approaches, where people use these Koopman type of ideas, where you say essentially, a lot of the system dynamics we see, in principle, a large number of nonlinear dynamical systems can be treated as nonlinear observations of higher dimensional linear dynamical systems. And so there's this sort of lifting process that people do, where you say, let's consider a higher dimensional, more complicated system that you're just seeing nonlinear measurements of, and you can explain a lot that way. And it has nice connections to these ideas of kernelization and featureization that I've also been kind of interested in, because I'm interested in too many things. But it's sort of that system identification, linear system identification, and model reduction from the control side, moving over to nonlinear system identification and Koopman type of ideas that the last 2 weeks of the class are about. And so it seems like this might be, this is an interesting and complicated enough set of dynamics that it might be a fun model system to try and apply some of those things to.
Yeah. Nowadays, a lot of the time, the problem is that the biologists tell us too much. Okay, and they're interested in all of the pieces. Okay, they say, we've just proved that this is happening. We've just proved that this is involved in the pathway from here to there. And so if you write down sort of a minimal model for anything that's been well studied, you've suddenly got yourself 12 state variables and nonlinear this, that, and the other. And they aren't interested in the question of which pieces can you throw out, right? They're interested in the question of what else is going on,
Right.
that we haven't identified yet, okay? So I think there's a lot of scope for, giving people ways of looking at, a lot of value in giving people ways of looking at these complicated models and say, in a systematic, not a trial and error, massive sensitivity analysis kind of way, but, some way of saying, what are the parts that matter? Which are the knobs that, so for example, I think for the last data you showed, Megan, what are the things that differ between one host and another that might actually explain these differences, and which differences are probably irrelevant because they can't actually, make things go differently. I don't think there's a lot of, well, here's how you do that out there. There's a lot of trial and error and seat of the pants.
Yeah.
So, I mean, these things, this is just to sort of say the kind of thinking you're doing strikes me as something where there's just sort of not a lot being done. And so that could be something very useful to get someone involved in.
Yeah. I would love to have a lower, a higher order linear model that would be computationally tractable. That would be fantastic.
Yeah.
All right. Chapter 1. Okay, well, I will take my leave. Thanks, Megan.
Thank you.